The human body is often compared to a high-tech universe, where countless processes unfold with remarkable precision. Within every cell, purposeful activity takes place, orchestrating the complex functions that sustain life. This intricate system operates seamlessly, relying on a delicate balance of chemical reactions and genetic instructions. As we embark on this journey, we'll explore how metabolism fuels our bodies and how genetics shapes who we are, revealing the extraordinary inner workings that define human existence. Glycolysis is a fundamental metabolic pathway that takes place in the cytosol of the cell. During this process, a single molecule of glucose is systematically broken down into 2 molecules of pyruvate. This breakdown releases a modest amount of energy, which is captured in the form of ATP and NADH. These energy carriers are essential for powering various cellular activities and set the stage for further energy extraction in subsequent pathways. Within the mitochondria, often referred to as the powerhouse of the cell, the tricarboxylic acid cycle, or TCA cycle, plays a crucial role in cellular energy production. As nutrients are broken down, the TCA cycle processes these molecules, leading to the generation of high-energy electron carriers such as NADH and FADH2. These carriers are essential because they transport electrons to the electron transport chain, where most of the cell's ATP is ultimately produced. By efficiently converting biochemical energy, the TCA cycle supports the cell's metabolic demands and overall vitality. After NADH and FADH2 are generated during earlier stages of cellular respiration, they play a crucial role in oxidative phosphorylation. This process takes place in the mitochondria, where these molecules donate their electrons to the electron transport chain. As electrons move through the chain, energy is released and used to pump protons across the mitochondrial membrane, creating a gradient. Ultimately, this gradient drives the synthesis of ATP, the primary energy currency of the cell. Oxidative phosphorylation is considered the grand finale of cellular respiration, producing the majority of ATP needed to power vital cellular functions. Cells have remarkable ways of adapting to low oxygen environments, a condition known as hypoxia. In these situations, they shift their metabolism to rely more on glycolysis, a process that breaks down glucose for energy without using oxygen. This leads to increased production of lactate, a byproduct that helps regenerate molecules needed for glycolysis to continue. At the heart of this metabolic shift is pyruvate, a key molecule that can either enter the mitochondria for further energy production when oxygen is available, or be converted into lactate when it is not. This flexibility allows cells to survive and function even when oxygen levels are limited. Hormones play a crucial role in regulating the body's energy balance. Insulin, for example, helps the body build and store energy by promoting the uptake of glucose into cells and encouraging the storage of nutrients. In contrast, glucagon acts when the body needs energy, triggering the release of stored fuels, such as glucose from the liver. This dynamic balance between insulin and glucagon ensures that energy is available when required and stored efficiently during periods of abundance. Together, these hormones maintain stable blood sugar levels and support overall metabolic health. Glycogen storage diseases, including von Gierke and McArdle disease, result from inherited defects in the enzymes responsible for glycogen metabolism. In von Gierke disease, a deficiency in glucose 6-phosphatase leads to severe hypoglycemia and the accumulation of glycogen in the liver. McArdle disease, on the other hand, is caused by a lack of muscle phosphorylase, resulting in muscle fatigue and cramps during exercise. These conditions highlight the critical role of balanced metabolic pathways in maintaining normal energy levels and overall health. When these pathways are disrupted, the consequences can range from mild discomfort to life-threatening complications. During fasting, the body undergoes a significant metabolic shift, relying less on glucose and turning instead to fat as its primary energy source. This process begins with lipolysis, where stored triglycerides in fat cells are broken down into free fatty acids and glycerol. These fatty acids are transported to the liver and other tissues, where they enter the mitochondria and undergo beta oxidation, producing energy. As fasting continues, the liver converts some of these fatty acids into ketone bodies, which serve as an alternative fuel for the brain and other organs. This adaptation helps preserve muscle protein and maintain energy balance during periods of limited food intake. Metabolic disorders such as phenylketonuria, maple syrup urine disease, and homocystinuria disrupt the body's ability to process certain amino acids. In phenylketonuria, the body cannot break down phenylalanine, leading to its accumulation and potential neurological damage. Maple syrup urine disease impairs the breakdown of branched-chain amino acids, resulting in toxic buildup and characteristic sweet-smelling urine. Homocystinuria affects the metabolism of methionine, causing elevated levels of homocysteine that can harm the cardiovascular and nervous systems. These conditions highlight the critical role of enzymes in amino acid processing and the importance of early diagnosis and management. Carbohydrate metabolism disorders, including galactosemia and hereditary fructose intolerance, arise from genetic defects that impair the body's ability to process certain sugars. In galactosemia, the body cannot properly break down galactose, leading to toxic accumulation that can cause liver damage, cataracts, and developmental delays if untreated. Hereditary fructose intolerance results from a deficiency in the enzyme aldolase B, making it dangerous for affected individuals to consume fructose, sucrose, or sorbitol. Exposure to these sugars can trigger symptoms such as vomiting, hypoglycemia, jaundice, and even organ failure. Early diagnosis and strict dietary management are essential to prevent serious health complications in both conditions. The urea cycle is a critical metabolic pathway that helps the body safely eliminate ammonia, a toxic byproduct of protein breakdown. In the liver, this cycle converts ammonia into urea, which is then excreted in urine. When enzymes in the urea cycle, such as ornithine transcarbamylase, or OTC, are deficient, ammonia can accumulate to dangerous levels in the bloodstream. This buildup can lead to serious health problems, including neurological damage and, in severe cases, death. Understanding the urea cycle highlights the importance of these enzymes in maintaining the body's chemical balance. Inside the nucleus, DNA replication unfolds during the S phase of the cell cycle. Here, the cell meticulously copies its entire genetic material, ensuring that each new cell will inherit an exact set of instructions. This process demands remarkable accuracy, as even minor errors can lead to mutations or cellular malfunction. Specialized enzymes work in concert to unwind the DNA, match complementary bases, and proofread the new strands. The precision of DNA replication is fundamental to maintaining the integrity of genetic information across generations. Gene expression begins with transcription, where RNA polymerase reads a DNA sequence and synthesizes a complementary strand of messenger RNA, or mRNA. Once formed, the mRNA is transported from the cell nucleus to the cytoplasm. Here, ribosomes attach to the mRNA and initiate translation, decoding the genetic information to build a specific protein. Transfer RNA. tRNA molecules bring the appropriate amino acids to the ribosome, matching each codon on the mRNA with its corresponding amino acid. The process starts at a start codon and continues until a stop codon signals the end of translation, ensuring the protein is assembled correctly. Chromatin structure plays a crucial role in organizing DNA within the cell nucleus. DNA wraps around histone proteins, forming nucleosomes that compact the genetic material and regulate gene accessibility. At the ends of chromosomes, telomerase is an enzyme responsible for maintaining telomere length, which is essential for cellular longevity. In most normal cells, telomerase activity is limited, leading to gradual telomere shortening and eventual cellular aging. However, in many cancer cells, telomerase becomes reactivated. allowing these cells to bypass normal aging processes and continue dividing indefinitely. Genetic inheritance follows specific patterns that help explain how traits and disorders are passed from one generation to the next. In autosomal dominant inheritance, only one copy of a mutated gene is needed for a trait to appear, often resulting in the trait being present in every generation. Autosomal recessive inheritance requires 2 copies of the mutated gene, typically causing the trait to skip generations. X-linked inheritance involves genes on the X chromosome, affecting males and females differently due to their distinct sex chromosome compositions. Mitochondrial inheritance, on the other hand, is passed exclusively from mothers to all their children, as mitochondria are inherited through the egg cell. Chromosomal disorders arise when there are abnormalities in the number or structure of chromosomes. Down syndrome, for example, is caused by an extra copy of chromosome 21, leading to developmental and physical differences. Turner syndrome affects females who have only one X chromosome, resulting in short stature and other distinctive features. Klinefelter syndrome occurs in males with an extra X chromosome, often leading to reduced fertility and learning challenges. Fragile X syndrome is linked to a mutation on the X chromosome and is a common inherited cause of intellectual disability. Vitamins play a crucial role in supporting blood health and nutrient metabolism. Vitamin C aids in the absorption of iron, which is essential for the formation of healthy red blood cells. Vitamin D helps regulate calcium levels, contributing to proper blood clotting and bone health. Vitamin K is particularly important for the blood clotting process, ensuring wounds heal effectively. Meanwhile, vitamins B12 and folate are vital for the production and maturation of red blood cells, helping to prevent anemia and support overall metabolic function. Anemia is classified into different types based on the size of red blood cells and underlying causes. Microcytic anemia, often resulting from iron deficiency, is characterized by smaller than normal red blood cells. In contrast, macrocytic anemia is typically caused by vitamin B12 deficiency, leading to larger red blood cells. Ferritin serves as an indicator of the body's iron stores, while transferrin is a protein responsible for transporting iron in the blood. Understanding these distinctions is essential for accurate diagnosis and effective treatment of anemia. Heme synthesis is a complex process that takes place in both the mitochondria and the cytosol of cells. The pathway begins in the mitochondria, where the initial steps occur, then continues in the cytosol, and finally returns to the mitochondria for completion. Each step is carefully regulated and involves specific enzymes. When there are defects in these enzymes, the process is disrupted, leading to the accumulation of toxic intermediates. This disruption can result in porphyria, a group of disorders characterized by dramatic clinical symptoms such as abdominal pain, neurological disturbances, and photosensitivity. Bilirubin is a yellow pigment produced during the normal breakdown of red blood cells. In the liver, unconjugated bilirubin, which is not water-soluble, is taken up by liver cells and converted into conjugated bilirubin through a chemical process. This conjugated form is water-soluble and can be excreted into the bile, eventually leaving the body through the digestive system. When the liver is unable to process or excrete bilirubin efficiently, levels of unconjugated or conjugated bilirubin can build up in the bloodstream. This accumulation leads to jaundice, a condition characterized by the yellowing of the skin and eyes. The intricate relationship between metabolism, genetics, and biochemistry highlights the delicate balance required for our bodies to function properly. Even minor disruptions in these complex systems can cascade into significant health challenges. Understanding these connections not only deepens our appreciation for the body's inner workings, but also underscores the importance of early detection and intervention. As research advances, it becomes increasingly clear how small genetic or biochemical defects can have profound effects on overall health.